When I am talking about cytokinesis, you should remember that in case of plant cells there is a cell wall, and in case of animal cells there is no cell wall. So this process of cytokinesis must be different in both type of cells. In plant cells it is different, animal cells the process is different. For example, if I am trying to explain you the process of cytokinesis, this event of cytokinesis in case of animal cells, so we have already seen the image that a cleavage furrow is formed. And this cleavage furrow formation is done by some actin and myosin proteins. Which proteins? Remember, actin and myosin. To your level, should just remember that these two proteins, these filaments, they come closer and they slide over each other. The sliding of this actin and myosin filaments eventually form cleavage burrow. This is reducing the diameter, and finally we have two cells, right? But in case of plant cells, you all know that we have a cell wall. Now what happens in case of plant cells? The cell dividing material, the cytoplasm dividing material is eventually present in the center. It collects in the center from what? From Golgi apparatus. There is formation of phragmoplast. What do we call it as? We call it as phragmoplast. It is nothing but the secretion of vesicles from Golgi apparatus. It starts from the center and it reaches the periphery. Then a cell wall is formed. There is cellulose, there is pectin, and then a plant cell divides into two. So in case of animal cells, the cleavage furrow starts from the top and meets here in the center and divides the cell. But in case of plant cells, the cleavage furrow or the division begins from the center, because here is some material which is called phragmoplast, which is formed from which apparatus? Golgi apparatus. It then reaches the top. So this is the difference between the process of cytokinesis between plant cells and between animal cells. So now I will show you an animation which is going to help you understand what is this process and how does this moves on. So this is how cytokinesis is going to proceed. So as you know that cytokinesis is the division of cytoplasm, and a cleavage furrow is formed in case of animal cells which divides the cell into two daughter cells. Now how this cleavage furrow is formed? Just look at it very carefully. There are some actin and myosin filaments. These are actin myosin filaments. When they slide past each other, the diameter you see is decreasing. Now the diameter is decreasing, and eventually when they meet at a point at the center, this cleavage furrow finally divides a single cell into two daughter cells, right? So in case of animal cells, which is responsible for this? Actin and myosin filaments. But in case of plant cells, plants have a special cell membrane. So the cell dividing material is in the center. This is Golgi apparatus. It is releasing some vesicles. These vesicles are forming phragmoplast when they arrange in the center in the form of microtubules, and then eventually a cell plate is laid. And when this cell plate is formed, this cell plate goes towards the cell membrane, and when the cell membrane is there is some release of cellulose from the vesicles. The cellulose is released, and after that there is pectin which is released, right? So after the release of pectin, after the release of cellulose, eventually two separate cell walls are formed which divide a single plant cell into two daughter plant cells. So you have now come across a difference in the process of cytokinesis between an animal cell and a plant cell. Since animal cell has no cell wall and plant cell has a cell wall, the cytokinesis process is different in both the cases. In case of animal cells, we have talked about actin and myosin filaments. When they slide against each other, they form a cleavage furrow which is starting from the top. The cleavage furrow begins at the top and meets at the center, and actin and myosin filaments are responsible for the division of cytoplasm. But in case of plant cells, since there is a cell wall, the dividing material starts from the center of the cell, which is phragmoplast. This phragmoplast is formed as a result of release of vesicles from which apparatus? From Golgi apparatus, remember. So eventually there is formation of cell plate, and then cellulose deposition and pectin deposition. From where the cellulose is coming? The vesicles which were released from Golgi apparatus, they contain what? They contain cellulose. They release cellulose in the center, they release pectin, and then eventually two different cell walls are formed which divide that plant cell into two different cells.